Hello students, welcome to LEELA'S tutorial. Today we will discuss about preparation of plant tissue culture media. In previous days they used to grow the plants or culture the plants by using the plant cuttings. So these cuttings were allowed to grow in soil or in liquid using fertilizers such as nitrogen, phosphorus, potassium and certain vitamins. If they were growing only using liquid or the water, it was referred as hydroponics. Right? There they have not provided any sucrose or any amino acid because the plant part itself has an ability to produce its own food. Right? Whereas here in plant tissue culture, apart from the fertilizers, that is nitrogen, phosphorus and potassium, you are supposed to provide sucrose and also amino acids because the cells are in shock. Right? So here let us see how we are going to prepare the plant tissue culture media. So here already in last class we have discussed that the scientist Haberland is the father of plant tissue culture, where he was successful in culturing the cells in Knobs solution, Knobs salt solution, and he was successful in producing the complete plantlet. Right? So here let us see, normally the plant tissue culture, they have first initially used all the nutrients in the solution form, that is sucrose, glucose, vitamins and fertilizers, and they were successful in producing the plantlets. Right? Then after that, here they were able to cultivate the whole plant or root culture or callus culture. Okay, so now we will see how we are going to prepare this media. We know what are the macro elements, micro elements. In last class we have discussed about the basic nutrients required for plant tissue culture. That is the essential elements such as macro elements, micro elements, vitamins, proteins, amino acids. Right? So all these, apart from the hormones or any inorganic supplements, we have learned what all are the compounds which must be added to our media. Right? So let us see how we are going to prepare the media now using all those compounds. So first we will see how we are going to prepare 1000 ml media. That is 1 liter media you are supposed to prepare. So for that, first you are going to take the double distilled water which has to be sterilized. So you are going to take sterilized double distilled water. You are going to measure 900 ml. And to that 900 ml, the media powder, that is all the essential nutrients. Already we have discussed in last class. All the compounds which we have mentioned, the macro elements, micro elements, everything must be powdered and it is added to this sterilized double distilled water. Right? To that 900 ml, you are going to stir till it is dissolved. If there are any precipitate or any sediments, you are going to heat them. Right? So by gentle heating also, the solution will become clear. Everything will be dissolved. So heating is done only if required. Okay? Then after that, you are going to add all the elements, the heat stable elements. What are the media powder is that the nutrient media and all other components. Right? So the complete macro elements, micro elements, sugars, vitamins, oxygen, cytokinins, etc. So everything you are going to add. Right? And then you are going to make up this 900 ml solution to 100 ml. You are not going to add 100 ml, 900 plus 100. You are going to make up because you have added these compounds. There may be increase in this 900 ml now. Right? So you are going to make up this solution after dissolving all the compounds. Right? Then you are making up to 1 liter. Once you make up to 1 liter, you are going to adjust the pH either by using NaOH or HCl. So now all the compounds, now you have a complete nutrient solution. So to this nutrient solution, if you want to prepare a solid, semi-solid media, you are going to add the gelling agent. If it is a liquid media or you want to go for a suspension culture, you can avoid this addition of gelling agent. right? So gelling agent, either you can make use of agar or you can make use of agarose, right? The purified form of agar is agarose. So here, before adding this gelling agent, after adding this gelling agent, you are going to heat it gently so that it will be dissolved and you get a clear solution, right? Otherwise the lumps may be formed. In order to avoid that lump formation, you are going to gently heat it, obtain a clear solution, then you are going to go for your steam sterilization process, that is autoclaving. So autoclaving is done at 121 degrees centigrade, 15 lb pressure for 15 minutes, so that all the contaminants present in the media will be sterilized or removed. Then here, now you are going to cool it, okay, not cooling completely because you have the gelling agent, right? You are allowing it to come down, right? The temperature has to come down. Before solidification, you are going to add your heat labile compounds. So in last class we have discussed, you are going to add either the coconut milk or tomato juice, right, orange juice or yeast extract or anything. So here these are heat labile compounds, that means these are heat sensitive compounds. They get denatured to the high temperatures. If you are adding before and you are autoclaving, all the compounds, the protein or the heat labile or heat sensitive compounds present in the media will be denatured. They will lose their activity, right? So in order to avoid that, the heat labile compounds must be added after autoclaving of the media, right? So again, the heat labile compounds may also be contaminated. So what you do, you have to sterilize the heat labile compounds also. Here you can't go for any heat sterilization, dry heat or the moist heat sterilization, right? Because we said that the compounds may get denatured. So what you are going to do, you are going to make use of filters. That is, the filter sterilization is done in order to avoid denaturation of heat labile compounds. So after filter sterilization, you are going to collect the filtrate and then add it to this autoclaved media. So after adding that, you are going to mix it well, and before solidifying, you are going to pour it into tissue culture flask or tubes or the petri plates, in whatever you want to culture, right? So once you maintain this, everything has to be done after autoclaving, everything has to be done in laminar airflow chamber, right? So under sterilized conditions, you are going to pour the media into the flask, right? Close them with the cotton plugs. Everything must be sterilized flask, sterilized cotton plugs, everything, right? So it must be sterilized. Then here in the laminar airflow, the airflow is on and you are allowing them to set. That is allowing them to get solidified because you are adding the gelling agent. So once they get solidified, right, you are going to close them with the cotton plugs, okay? And now after that, you have to sterilize your explant, whatever explant you are going to take, right? Explant is nothing but any part of the plant body which you are choosing to culture, right? If I am taking a meristem or a leaf, what I am going to do, I have to go for the surface sterilization of the explant. After surface sterilization of the explant, I am going to inoculate that into the media. So after inoculation, it is in incubation at favorable temperatures so that I can produce a complete plantlet using the explants, right? So here this is the media preparation procedure. If you have any doubts, put it in comments and subscribe for further videos.